Hello. Hi. Am I audible? Yes. Hello. Okay. Okay. Hi, everyone. Hi. Good evening and welcome back. Yes, I hope I'm audible. Yes. Okay, so welcome back to yet another class. I'm Alka from Gosko Learning. We are the fastest growing edtech in GCC, and our aim is to provide quality education for all. Yes, welcome back. Happy to see you all again. So as you all know, we have been going through the heredity, right? Okay, I think we are having some glitches. Is everything okay? Is the audio fine? Hi, hello. Maybe can you connect to the network? 3. Yes. We have audio issues. Hello guys. Are we back? Is the technical issue resolved? Am I audible now? Can you please comment? Can you hear me? Can you hear me? Yeah? I hope the issue is solved. Yeah? Yes. Yes. Okay. Okay. I guess we are fine now. We are fine, right? Okay. 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 Sorry for the inconvenience. That was a minor network issue. Sorry about that. Yes, I'm back. Okay. Again, we are here to complete our 8th chapter Heredity. And as this is the final session of this chapter, I hope you all are eagerly waiting for this, for the climax part. We have started it like 2 weeks before, and yes, today we are ending it up. And as you all know, we have covered pretty much topics here, right? We have covered up pretty much topics. Just let me go through the content again so that you all will get to have an idea. So yes, these are the topics we are almost to cover up. We have covered up the terminologies, Mental and his contributions, monohybrid, dihybrid cross. Until that, it's done. So we have some laws of inheritance and a sex determination part, which is yet to be finished. Right? Okay. It's yet to be finished. Hi, hi, hi. Hi, all. Hey. Yes. Okay. So, as you all know, the major content of this chapter comes under this mono and dihybrid cross, along with the sex determination part. So, sex determination in human beings is a major content in this chapter, or a topic where you have to focus a bit more, because that's the part where normally questions have been asked, and we can expect, helpfully and hopefully we can expect questions from that part. So today we'll be covering up those portions. Yes, thank you all for coming up in the live. Everyone, hi there. Hey, yes, yes, yes. So, and in every class, we normally give a small tip for your learning things, right? So today also I've got a small tip, and the tip is nothing. Whenever you are there to learn something, whenever you are preparing for your boards or your pre-boards, make sure that you are spacing out your study area. Make sure you are not in between a crowd or in front of a television, making your auditory in somewhere and your visuals in somewhere. So whenever you are ready to learn, make sure that you have created an atmosphere which is good to go. So always space out your study area, always space out from the crowd and noises. Make sure you're able to focus. And of course, spacing out is one point, and the second thing will definitely be distractions. So minimize the distractions. I know it will be very difficult. Distractions are there in our pockets all the time, right? We have that small mobile, we have one tablet on the table, or a laptop from which you are studying. So Distractions are there, but try to minimize the distraction when you are in a mood to study. And of course, the third point is take breaks. Take breaks. No human in this world can focus in something for more than 45 minutes. The maximum you can focus on learning, or let it be anything, let it be any sort of activity you're doing, more than 45 minutes, our brain doesn't do it. It gets exhausted. So make sure you are taking breaks in between the study. Don't just rush in the night just before your boards and try to cover up the syllabus. It won't work. It won't work. It will just get you exhausted and your exam will turn out to be big, ugly thing. So better take breaks in between your studies and make sure you have enough time to take breaks so that you start priorly. Your preparation start priorly. So that's the point, that's the tip, that's the learning tip I want to share with you. So always make sure that you're spacing out whenever you're ready to study. Make sure your distractions are less. Make the distractions in silent mode or switch it off, or whatever the distraction is, switch it off, turn it off, minimize it for the moment you're studying. I'm not saying to take it away from your life, it's impossible. And then, of course, take breaks. Take breaks. At least in 45-minute intervals, just take a break, go just walk around. And the break is not supposed to be any blue screen media. Okay, it's not the mobile or laptop or the computer that you have to take breaks in. So make sure you go out, you are in the nature, maybe get some fresh air, drink some water. That's the break. So yes, that's the tip I want to share, and I hope now you are ready for the session. Yes, hi all. Yes, yes, yes. Very happy to see the comment session with lots of hearts and loves and fires and high fives. Yes, thank you so much for your love. Yes, yes, hi. Take a break and have a Kit Kat. No, don't do that. Now don't take the break. Now you focus. Okay, so I hope you have these things in your mind. We have studied a lot of stuff in this chapter. We have learned too much terminologies, genes, alleles, genotypes, phenotypes, homozygous, heterozygous. That's where we started the chapter. Then we have seen Gregor Johann Mendel, that great scientist who actually led us to the world of genetics, to the heredity part, right? And then we have seen what a monohybrid, which is taking one character and studying its inheritance, and then dihybrid, which is taking two characters and studying its inheritance. So that's where we stopped in the previous class. So let me begin with giving a small intro from the dihybrid. I still believe that it's there in your brain. Your cerebrum hasn't cheated you yet. I hope so. Have your cerebrum cheated you yet? No, right? It's there. It's stored in there. Okay, I hope it's not still in your amygdala. It's stored for a long time. I hope it's in your long-term memory. Okay, okay, fine. So as we have already seen, the dihybrid cross is where we take two different characters. In the previous class, we have taken the color of seed and size of seed, right? We have seen the color of seed and size of seed and the shape basically, right? The green and yellow color, round and wrinkle shape. That's the character we chose. That's the characters we chose, right? And finally we have came to a conclusion that in a dihybrid cross the phenotypic ratio will always be 9:3:3:1, and we have found it, right? So whenever you get a question from the dihybrid cross ratio part, mm, just copy paste this ratio in your brain, because we'll need it. If it's a monohybrid cross, the phenotypic ratio will always be 3:1. If it's a dihybrid cross, the phenotypic ratio will always be 9:3:3:1. Done. Okay, fine. And we have done this question. I hope you all remember it. Today I'm here with a new question, which is from the dihybrid cross part, and let's begin. I hope you all have a book and paper with you, and we'll begin the class. Okay. So this is the question. This is a PYQ, and as you can see, it's a bit complicated. The question itself seems much big, right? Okay. So this is how the question goes. In a mice, black cod color is dominant over the brown cod color, and the solid pattern is dominant over the white spotted color. And spotting are controlled by genes that assort independently. Okay, that's a long story. That's a long story. Let me break it down to you. It's simple. There are two types of mouse here. Okay, there are two types of mouse. So the one mouse is black cod color. Its cod color is black, and they have already given it as black mouse uh, over brown. So we have black mouse and brown mouse, and their cod color, the texture is different. One is having a solid pattern, the other is having white spotted pattern. So this black mouse is actually having solid pattern. So it is a black solid mouse. The other one is brown cod, which is having white spotted. Okay. So these are the two characters we have taken here. It is white spotted actually. Not a solid pattern, but a spotted. There are spots on the fur. That's it. So now we are taking a cross. This is the part to focus. Let's see the question. The cross is a homozygous black spotted mouse. Okay, so how is the cross? A homozygous black spotted mouse is crossed with a homozygous brown solid mouse. So that's the cross. So let's focus on the part where the characteristic cross is given, and now let's try to make that cross. Okay, so whenever you get a question from the dihybrid or monohybrid cross, make sure what the question is asked for. And the first part, they explained the genotype and phenotype of the mice. Okay, let it be like that. Now the question part is just the last sentence. That's where we have to focus. What have they asked? What have they asked? A homozygous black spotted mouse. So we need a black spotted mouse which is crossed with, what is it crossed with? It is crossed with a homozygous brown solid mouse. So brown solid, brown solid. Yes. I think the question is clear now. We can easily take a cross of that, right? We can easily take a cross of that. Let's take the cross. Black. What is the letter for black? It's already given in the question. It's a dominant trait. So you have to choose capital B, capital B. That's the black. Now, spotted. What is the letter for spotted? Where is it? Where is it? So, over white spotted. The letter taken for spotted is small s. So, it is capital B, small s, which is crossed against. It is crossed against what? It is crossed against brown solid mouse. And brown, I think it's already said. See, the brown cod color is small b. So, it is brown means small b, and what is it? Solid mouse, solid. The pattern, the skin pattern, the cord pattern is solid, which is capital S. It's already given in the question. The solid pattern is capital S. You don't have to pick a letter, it's already given. So this is the cross that the question is talking about. Now this is the first question from the paragraph, or this is a case study like of question where you have to solve it from the case given. So this is the case, and the question is this: the genotype of the parent is dash. I think from the question it's clear, and we have already arrived at a conclusion. Genotype of the parent is capital B, smallest, which is crossed against small b, capital S. So I think that goes to option, yeah, option B. Capital B, smallest, small b, capital S. So that is option B. I hope you got it. So just read the question. It's a case study like a question. Simple. It's not done. The question is not done. From the same question, next thing is asked. What is the next thing? Select the incorrect statement regarding the F1 progeny obtained. Okay, so now we have to talk about the F1 progeny. So to talk about F1 progeny, we actually have to make a cross, right? So this is the parental trait. Now to get the F1 progeny, they're asking about F1. Select the incorrect statement regarding F1 progeny. To talk about F1 progeny, we actually need to know which the F1 progeny is, right? So we have to talk what the phenotype and genotype of F1 progeny. That's what the question is about. So let's do the F1 progeny first. Let's find out the F1 progeny. Okay, so I think you can easily find the F1 progeny from this question, right? We have already done the round yellow and green wrinkle seed. It's the same thing. Instead of round and yellow, we have black, spotted, and brown solid. That's it. Two characters, dihybrid. That's it. So let's see, this is the parental trait. Now let's take the gametes. How would you take the gamete? How would the gamete look like? So this is a homozygous parent, right? So as you can see, it will create gamete capital B, small s. This will be the gamete from the first mouse, which is the first parent. And the second mouse, let it be male or female, we don't know who is the male or female here, but the second parent will be having again small b, capital S, small b, capital S. Since the parent is showing homozygous nature, there will only be one kind of gamete. There will only be one kind of gamete because the parents are showing only homozygous nature. There is no heterozygosity here, right? I think you understand that. Yes. Yes. Hi all. Okay. Now the F1 generation. So since we are talking about the F1 generation, I think it's clear now. It's clear now. If you have got the gametes, the male gamete fuses with the female gamete and it gives a baby, right? It makes the zygote. So how would the baby look like? How would the baby look like? What would be the genotype of baby? Please comment it. Please comment it. Yes. How would the baby? What would be the F1 generation be like? Yes. Yes. Yeah, it's clear only, right? It's very simple, right? What is it? The baby would be capital B, small b, capital S, small s. Capital B, small b, capital S, small s. So what would the baby look like? What would this baby look like? This will be a very teeny tiny mouse which is black colored because capital B will be dominating small b, so the color will be black. And here capital S will be dominating small s. So it will definitely be S. What is S for? What is the S for? This was solid pattern. Capital S is solid pattern. So it will be a black mouse with solid pattern. So it will be a pure black colored teeny tiny baby. Cute baby, right? Yeah. So this is the question about. So the F1 generation will be a black solid mouse with capital B, small b, capital S, small s pattern. What was the F1 generation? Capital B, small b, capital S, small s. This is the baby. Now let's read the question again. This is the second question. Select the incorrect statement regarding the F1 progeny. Okay, let's read it. A. Phenotype of all mice of F1 generation is brown coat with solid pattern. Is it true? No, it is not at all brown coat. It is black coat. So wrong statement. I think we got the answer, right? Let's read the other sentences. Phenotype of all mice of F1 generation is black coat with solid pattern. Black coat with solid pattern. Yeah, it's true. True, right? It's true only, right? Yes. Then, genotype of all mice of F1 generation is capital B, small b, capital S, small s. True, right? It's capital B, small b, capital S, small s. True. Genotype of all mice of F1 generation is capital B, capital B. No, wrong. That itself is wrong. You don't have to complete it. So if you got the first statement wrong and you stopped it right there, then you have lost the score. Because they have actually given two incorrect statements. I think in the question they have given it like that only. There are incorrect statements. There are two statements. So you have to choose it. So which are the wrong statements here? It is option A and option D. Because the F1 generation is capital B, small b, capital S, small s, which is a black colored, solid, teeny tiny baby. Simple. Okay. I hope that's clear. Show the test screws of the capital B, small b, capital S, small s. What is the test cross about? Test cross is nothing. When you are making a cross with the homozygous recessive trait, that is called a test cross. What is it? What is it? Test cross is nothing. It is a cross which is done with a homozygous recessive trait. Homozygous recessive trait. What is that? What is that? Homozygous, both the alleles are same. Recessive, both the alleles should be in small letters, right? Both the alleles are hidden, or you can say it is submissive, right? Okay, so let's make a cross. That's the question. That's the C part of the same question. Let's try to do it. It's simple only. So we have to cross capital B, small b, capital S, small s with a test cross. So what will be the test cross like? It is homozygous recessive. So it will be small b, small s, small s. That's a cross. That's a cross. Simple, right? Simple, right? Any issues? Clear? Okay. Okay, I hope it's clear. Fine. Okay, fine. So this will be the parental trait. Now let's take the gametes. Let's take the gametes. So how would the gametes be like? Unlike the other case, I think we will get much gametes here, right? So the gametes would be capital B, capital S. This will be one gamete, and we can get capital B, small s, another gamete, small b, capital S, another gamete, small b, small s. That's another gamete. So four types of gametes are possible from one parent itself. So it's a long story, which is crossed with capital B, small s. This is the only possible gamete from the test parent, right? Because it's a homozygous parent, only one type. So yeah, you can make crosses with all of this and you will get F1 generation with all these characteristics. So how would it be like? The F1 generation would be looking like, if this gamete is fusing with this gamete, the baby would be capital B, small b, capital S, small s. So that's a black colored solid mouse, right? Then if this gamete is fusing with this gamete, then it will be capital B, small s, small s. So how would that be? That's a black colored spotted mouse, black colored spotted mouse. Then if this gamete is fusing with this gamete, then it would be small b, capital S, small s. Am I right or not? Am I right or not? It's right, right? Okay, so it's small b, capital S, capital S, small s. Yes. So it is brown colored solid mouse. Okay. Then if this gamete is fusing with this gamete, it will give a baby that is small b, small b, small s, small s. That is brown colored spotted mouse. Right? Right? So it is possible to get all these phenotypes in the F1 generation, and this is how the test cross would look like. This is how the test cross would look like. Simple. So that was a case study based question, which has three subdivisions in it. So the question itself is simple, but you have to use a bit logics here and there. That's it. Yes, I think that's clear. If then let me move on to the new topic, that is laws of inheritance. Yes, laws of inheritance is the topic that we have to talk now. Yes, I hope there is no issue in the comments. Yes. Okay, fine, fine, fine. Moving on. Principles or laws of inheritance. Principles or laws of inheritance. And who proposed these laws? These proposed by, these laws are proposed by none other than Gregor Johann Mendel, the father of genetics, right? So it is proposed by Mendel, Gregor Johann Mendel, who is known as the father of genetics. And he has proposed mainly three laws. The first law is known as law of dominance. Second is known as law of segregation, or it's the second law is also called the law which is talking about purity of gametes. Purity of gametes. Yeah. The second law is also known as purity of gametes. And you will know why it is so. And then the third law, which is law of independent assortment. So these are the three laws or principles of inheritance, or you can say laws of inheritance. It's a very simple concept. Then and there you may get the one word questions, or not the one word, the one mark questions like define the law, or like they might give a monohybrid cross and ask what is this law about, or what kind of law can you conclude from this cross. So if it's a monohybrid cross, you can arrive at the law of dominance. If it's a monohybrid cross, you can arrive at the law of dominance, even law of segregation. And if it's a dihybrid cross, you can arrive at a conclusion of law of independent assortment. So normally they give the crosses and ask which law is connected here, or which law can scientists conclude from this cross. Like that, you can expect questions like that. Okay, so let's learn the laws. The first law is law of dominance. It's a very simple concept, nothing. Characters are controlled by discrete units called factors. Okay, what is that? What is a factor? That's the first time we are hearing that term, right? Okay. So characters are controlled by discrete units called factors. Factors occur in pairs. Okay. In a dissimilar pair of factors, one member of the pair dominates the other. Fine. Now, factors are called genes. So that's it. So the factors which are talked here, it's about genes. It's now that we have actually got a term gene. In the older days, in those times, there were nothing. We didn't know. The scientists, they never knew what genes were. So they just used to call it as factors. That's what the genes were called on those days. That's it. It's the same stuff. It's the same thing. Okay. So characters are controlled by discrete units called genes. So every character is there in your gene, right? If it's your height is the character, it is controlled by capital T, which is a gene, or capital T small t, or a small t small t. So these are the genes, right? And factors occur in pairs. Yes, the genes are always in pairs. It comes in pairs. Yes. In a dissimilar pair of factor. Okay, what is a dissimilar? This is not a dissimilar. This is not dissimilar. So this is a dissimilar pair. In capital T, small t, they are dissimilar pair. So in a dissimilar pair of factors, one member of the pair dominates the other. So the dominating one is the dominant gene, the other one is the recessive gene. So if you take the case of capital T small t, if you take the case of capital T small t, in that dissimilar pair of genes, you can see that capital T always dominates the small t. One member dominates the other. So small t is known as the recessive gene here, and this is the dominant one, right? So now we are using an extra term called alleles. We know that this contrasting pair of the same gene is known as alleles. So this is the recessive allele and this is the dominant allele. That's it. That's what the law is talking about. And yes, now factors are called genes. This is the law of dominance. So whenever law of dominance is asked, make sure you have a definition for it. You know how to define it. Just keep this capital T and small t in your mind and just define it. That's it. You can use the same terms. Done. Moving on. This is a question which is based on the first law of dominance. Or let's see if we can answer the question. In a monohybrid cross, one of the allele is dash and the another is dash. Yeah, simple. Simple. What's the answer? In a monohybrid cross, one allele is dash and the another allele is dash. One is dominant and the other is recessive. That's the answer. One allele is dominant and the other is recessive. The other is recessive. Yes, that's it. That's a simple question. A 1 mark question. Now comes the law of segregation, and this is the law which is also known as the purity of gametes. This law talks about the purity of gametes. So let's see what the law is about. If I read it, it's something complex like this. During gamete formation, the factors or alleles of a pair segregate from each other. One gamete will receive one factor and another will receive another factor. That's the law about. So if I want to put it or break it down to you, it's simple like this. Suppose a gamete is being formed, and from where the gamete is formed? The gamete is being formed from our own body parts, from our gonadial parts, right? Okay. So when a gamete is being formed, suppose it is capital T small t. It is from the capital T small t that the gametes are being formed. If that's the case, the factors or alleles of a pair, these are the alleles of a pair. Capital T is one pair of the alleles and small t is one pair of the alleles, right? So in alleles of a pair, they segregate from each other. They separate when gamete is being formed. in a pair of alleles, they segregate from each other in such a way that one gamete will receive one factor and another will receive another factor. Simple. So you know that if we have a gene like this in our body or in a plant's body, in an organism's body, when a gamete is being formed, what happens? When a gamete is being formed, these alleles separate so that one gamete would receive only one pair of the factors. Only one of the pairs. Only one of the pairs. That is, only capital T would go to one gamete and small t would go to another gamete. Simple like that. When a gamete is being formed from a pair of alleles, one of the pair goes to one gamete and the other pair goes to the other gamete, which means this thing is not possible. If this is a gamete, capital T and small t cannot be in the same gamete. This is impossible. That's it. But this is possible. One gamete can receive only one of the pairs. Simple like that. That's it. That's the law talking about, and this is why it's also called purity of gametes. It's talking about purity of gametes means a gamete will always be pure with one allele in it. It can't be mixed up. That's the purity of gametes. I hope you understood that concept. Okay, and yeah, there are some adding on points like if homozygous parent produces all gametes that are similar, heterozygous parent produce two kinds of gametes. Yeah, even that's also understandable, right? Homozygous parent, suppose it is capital T or small t, homozygous parent, they produce all gametes that are similar. This parent will able to produce only one kind of gamete. No matter how many gametes it produces in its life, all the gametes will have capital T in it. That's it. And if it's a heterozygous parent, that is, if it is capital T, small t parent, this parent can produce two kinds of gametes. Which are they? One gamete will be capital T and the other gamete will be small t. Understood? It's simple. Homozygous parent produce all gametes which are similar. Heterozygous parent produce gametes which are dissimilar, or maybe two kinds of gametes. That's it. Simple like that. Got it? Yes. Yes. Hello. Hi. Hey. Are you all saying hi to make sure that I'm here only? Yes. Hi. Okay. So we have the third law, that is the law of independent assortment. Law of independent assortment. We have covered up law of dominance. We have covered up law of segregation or the purity of gametes. And then this is the last law put forward by Gregor Johann Mendel, that is law of independent assortment. Simple. Simple. It's nothing. It's nothing. It's just talking about dihybrid pairs. I'll first show you the example, then I'll explain the law so that it would be very simple to you. It's nothing. Suppose it is this capital R, small r, capital Y, small y thing. What is this? This is round yellow colored plant, right? A plant, a pea plant which is having round seed which is yellow colored, and that is heterozygous. Okay. If this is the condition of a plant, or if this is the condition of an organism, the law of independent assortment states that when two pairs of traits are combined, so which are the pairs of traits here? We have round, which is one trait. We have yellow, which is another trait. One is the shape, other is the color. That's the two traits here. Okay. So one is the round, one is the round shape, the other is yellow, which is the color. So shape and color are the two traits which are selected here. Okay, this is what the law of independent assortment is talking about. When two pairs of traits are combined in a hybrid. Hybrid mean what? One parent's gamete, another parent's gamete, it fuses, it forms a hybrid. That's it. So when it is combined in a hybrid, segregation of one pair of character is independent of the other pair of character. Segregation of one pair of character. Okay, I'll write it again. This is capital R, capital Y, small y. Capital R, small r is one character, capital Y, small y is another character. So the law states that segregation of one pair of character is independent of the other pair of character. That is, capital R, small r segregates itself. It doesn't have to wait for capital Y, small y. So their independent assortment means one character, that is shape, is independent of color, which means from this parent any kind of gamete can be formed. The capital R can go with capital I independently. Capital R doesn't have to wait for small r to come along with it or small y to come along with it. They are independent. Capital R can go with capital Y if it wants. Capital R can go with small y if it wants. Small r can go with capital Y if it wants. Small r can go with small y if it wants. Simple like that. It's very simple. What is it? Whenever a hybrid is formed, the assortment, whenever a hybrid is formed, the assortment of one pair of character is independent of other, which means color is independent of shape, or my hair color is independent of my skin color. It doesn't mean that all the skin colors should have same kind of hair color. No, whichever the hair color is, skin color can be anything. Suppose there is a person who is having black color. That person can have a skin color which is very pale. Suppose there is another person with black hair and mid-toned skin color. It is possible. So one character's assortment is completely independent of other character. That's what the law of independent assortment is stating. I hope it is clear now. Whenever you hear that term independent assortment, just put this thing. Just put this capital or smaller capital Y, small y in your brain, so that you will be easily able to explain the law in words. Okay. So that's it. These are the three important laws. It's a very important part in our chapter also. I hope it stays in your brain. I hope you keep revising it until your, at least until your pre-boards and boards. Okay. So that's it. That's the most important part which is just covered up. Now, the last portion of the chapter, which is sex determination. This is the last portion of the chapter, which is sex determination. First, we'll discuss about sex determination in humans. Then we'll go to sex determination in some small creatures. Okay, lower group of organisms. Both are important. Both are equally important. There have been questions from both the part. So, sex determination and the crossing part is the major part, major topic of this chapter. Okay. So, the first one is sex determination in humans. So, what is this thing talking about? What is the topic about? The topic is very simple. Sex determination. How to determine whether a baby is going to be girl or a boy. That's the topic about. When a couple is having a baby, the newborn's sex, what is its biological sex? Who determines the baby is going to be a boy baby or a girl baby? That's the topic about. So, what is your idea about this? Who do you think can determine the sex of the baby, or it's completely supernatural? What do you think about it? As science students, you should have some conclusions, no? So it's completely, you can say most of the things are controlled by probability. It's purely chance. The sex of the baby is purely chance, but it is actually determined by one of the parent. It's either father or mother, right? So, whom do you think is determining the sex of a baby? The baby is going to be a boy baby or a girl baby? Okay, I think you yourself can answer. I'll start with the topic. The topic is simple like this. I hope you have an idea about the chromosomes in us, right? So, chromosome pairs present in the zygote determines the sex of the female and the male child. So, that's the answer. Basically, the chromosome determines what the sex of the baby is going to be. So father and mother contribute equal amount of genetic material. The equal, just highlight in it. Father is giving 50% of his chromosomes and mother is giving 50% of her chromosomes. So the contribution to a baby's body. My body has 50% DNA content from my father and 50% DNA content from my mother's body, right? So the genetic material available in a baby, in a person, is completely equally distributed. by both the parents. Okay. But each trait can be influenced by both paternal and maternal DNA. So any character in my body, let it be my let it be my skin color, let it be my eye color or shape of my nose, anything, any trait I have in my body can be influenced by whatever chromosomes both my parents have given. Because if one is dominant, that character will be expressed in me. If the other one is recessive, that will be staying hidden in me, which would be expressed in my babies or my baby's babies in a new generation, right? Okay, so let's come back to the topic. I think you have an idea about this chromosomes in the body. I'll just simply brief it up. So as human beings, Homo sapiens, each species have their own number of set of chromosomes and all. Okay, so Homo sapiens as humans have 46 chromosomes in each and every cell in our body. If you take a single cell in your body, it will have 46 chromosomes which occur in 23 pairs. So these 46 chromosomes are not alone, they occur in pairs. We have 23 sets of chromosomes and each set will have 2 chromosomes, right? So we have 46 chromosomes. Out of this 46 chromosomes, a part of them are called the autosomes. Autosomes. And the next part is called allosomes. Allosomes. Oh, these allosomes are also called the sex chromosomes. Maybe that word is more familiar to you. Sex chromosome. So we have autosomes in our body and allosomes in our body. All together, they will contribute to 46 chromosomes. So the autosomes are basically the 22 pairs. That means the first 22 pairs of chromosome in our body, which is 44 in number, right? So the 44 chromosomes in every human being is called autosome. It can be present in both male and female equally. The 1, 2, 3, 4 like that. We have 22 pairs. Those 22 pairs or the 44 chromosomes, they are called the autosomes. There are plenty of characters controlled in these chromosomes. Okay. Going on to allosomes or the sex chromosomes, these are just the remaining one pair. The remaining one pair of chromosome, or you can say 2 chromosomes. The remaining one pair or the 2 chromosomes, which are the 23rd pair. We have 23 pairs. Among that, 1 to 22 is called the autosomes and the remaining 23rd pair is called the sex chromosome or the allosomes. This allosome or sex chromosome is different in male and female. If it is a male, I think you know the concept clearly, maybe better than me. So, if it is a male, this is the condition. If it is a male, this one pair of chromosome will be occurring in XY condition. And if it is a female, this one pair, the last pair, the 23rd pair of chromosome in our cell will look like XX condition. That is nothing. There is no X and Y inside our gene. There is no alphabets in the gene. Why do we call it as X and Y, XX and all? We are calling it in such a name because of the appearance. That is, in females, both the sex chromosomes look equally, what do you call? They look similar. They have the equal content. Whatever the first X chromosome is having, the same content is repeated in the second X chromosome. They look alike. Their size is same. Okay. Now going on to the male, one sex chromosome is X and the other is Y, means Y is a little bit smaller. That's it. If suppose this is the X chromosome of male, the Y chromosome is a bit smaller in size. And the content, the genetic content of Y chromosome is also very low. When you compare with any other 45 chromosomes, that Y chromosome is very small in structure and it's also having very less content of characters in it. That's it. So this is how the chromosomes are arranged in our body. I think you can answer this question very simply. In human males, all the chromosomes are paired perfectly except one. This was a previous year question. In human males, all the chromosomes are paired perfectly except one. This or these unpaired chromosomes, chromosome E's or R, Which are they? A large chromosome, small chromosome, Y chromosome, X chromosome. Which one do you think? In human beings, all the chromosomes are paired perfectly except one. That exception is this Y chromosome. Y chromosome is the one who is unpaired, or you can say who is dissimilar to its pair because it's very small. That's it. Got it? Okay, moving on. This is another question from the same part. The number of autosomes in human zygote is. Number of autosomes in human zygote is. Very simple. The sperm will be carrying 23 chromosomes. The egg will be carrying 23 chromosomes. When they fuse, we get a zygote, and the zygote should have 23 plus 23 chromosome, that is 46, right? So the number of autosomes in human zygote. So out of this 46, there will be 44 autosomes and 2 sex chromosomes. So every zygote, let it be male or female, every zygote, if it's a normal zygote with no syndromes, if it's a normal human, it should have 44 autosomes. That's the answer. Moving on. How is the sex of child determined in human beings? Very important. Very, very, very important question. Very, very, very important question. I repeat it. So these questions have been asked in mostly all years, and they keep asking it in one or the other way. Anyways, the answer is this: human sex determination. That's it. So this is one question and this is another question, but both are asking for the same answer. One is, how is the sex of child determined in human beings? The other is, in humans there is 50% probability of the birth of a boy and 50% probability that a girl will be born. Justify the statement on the basis of mechanism of sex determination in human beings. Same question, same topic. If you know that simple thing, you can score in it. And the thing you have to know is this: so whenever such a question is asked regarding sex determination in humans, you basically have to depict a diagram, an illustration, which is very simple. Suppose it is a cross, just like we did a monohybrid cross and a dihybrid cross, this is another cross. So the parents are going to be male and female, right? So we will have a father and a mother. We are talking about humans, right? So the parent will definitely have the X, Y, and X chromosome. So this is the picture you have to depict for both the question. This is the answer for both the question. So the parent will be having XY chromosome and XX chromosome. Just depict it like that. So the parental gamete will be having X, so not the gamete, the parent body will be having XY and XX condition. And the gametes which will be formed out of this, the gametes which are coming out of it will be like this. Since male is heterozygous, in humans, the males are, the fathers, they are heterozygous. If the father, if a parent, if the organism is heterozygous, they can produce two types of gametes. We already learned it in the law of segregation, right? Law of segregation, in the law of purity of gametes, we have learned that if the organism has heterozygous condition, that is dissimilar alleles, if the alleles are dissimilar, it can produce two types of gametes. The first gamete will be X and the second gamete will be Y. And since it is a male human, this gamete is what you call as a sperm. That's it. And going to female, since the females in human beings are homozygous, no matter how many gametes they produce, all the gametes will have only one kind of sex chromosome, that is X. Simple. Simple. Now let's take the F1, or let's take the zygote being formed out of this. So when a zygote is being formed out of this, what will be the condition? The cross is simple. The X sperm fuses with the X egg, the baby or the zygote will be having XX condition in its body. That's it. If the Y sperm is fusing with the X egg during fertilization, during the fertilization process, if a Y sperm is fusing with the X egg, there is no need of saying X egg because all the eggs are egg. All the eggs are X. Yes. So if the Y sperm is fusing with the X egg, the zygote is going to be X, Y. Yeah. So there is just 50% probability for a baby to become XX and 50% probability for a baby to become XY. And that probability, it's up to complete chance, right? Which sperm is fusing with which egg, it's completely chance, nothing to do with any technicalities or any kind of biological things. Which sperm is coming in, no idea. So whatever the sperm is coming in, no matter what the genetic content of the sperm is, that is complete probability. Which sperm is fusing, it's the X sperm or the Y sperm, it's probability. But if the X sperm is fusing with the egg, it will definitely be a baby girl. And if the Y sperm is fusing with the X egg, it will definitely be a baby boy. So the chance of a baby being girl is 50% and chance of a baby being boy is 50%. So whenever you get a question from that part, just do this cross. Make sure that you have rewritten the technicalities about the chromosome number, autosomes, allosomes, what's the difference in male and female, and how this 50% is acquired. That's enough. If you are able to express that in a very good manner, that mark is yours. Just take it. Simple like that. So this was the question, how is sex of a child determined? So sex of a child is determined basically by the type of sperm that a father produces or the type of sperm which fuses with the egg. And in humans, 50% probability for girl, 50% probability for boy. Justify the statement. So to justify that statement, you basically have to draw the illustration. Just make that illustration and explain the chromosome things. That's it. So that's what the question is about. So this is the same question from the same topic. No, not the same question. It's a different question from the same topic. Two individuals are shown using geometric shape. So one is XFY, the other is XMXM. Their sex chromosomes are respectively denoted by XF, XM and Y. Okay. What are the possible combinations of sex chromosomes for their male and female offsprings respectively? Simple. So here we have a parent who is having XFY condition, another parent who is having XM condition. So I think from the picture itself it's clear. This is a female and that's a male from the picture. That's all we can identify. Let's see how the baby is going to be. We just have to find the sex chromosome of the male and female offspring. So you basically have to make a Punnett square. That's it. So let it be male and female like this. We have a male, we have a female. So XF, Y are the gametes of the male. XM, XM are the gametes from the other parent. We don't know who the male and female and all. It's not denoted in the question. And yeah, we also don't know whether they're talking about humans. Nothing is mentioned there. So let's just make the zygote. The zygote would be like this. The first would be XM, XF. The second would be XM, Y. Then we have XM, XF. Then we have XM, Y. So XM, Y will be the boy, or you can say that will be the male here. XM, XF will be the girl or the female here. So that's it. The sex chromosome. What are the possible combinations for sex chromosome for the male and female? So for the male, it will be XMY and the female, it will be XMXF. That's the answer. Simple. Done. So yeah, that's it. Now this is a assertion and reason kind of question, which is also very simple. Nothing much to think too much here. It's simple. Sex of a child in human beings will be determined by the type of chromosome he or she inherits from the father. Yeah, it's true or false? It's true only, right? Sex of a child will in human being will be determined by the type of chromosome he or she inherits from the father. True. A child who inherits X chromosome from the father would be a girl. True. While a child who inherits Y chromosome from the father would be a boy. Yeah, that's also a true statement, and it completely explains the assertion. So yes, assertion and reason are true, and reason is the explanation of assertion. So that will be option A. Moving on, this is the last topic, sex determination by the environment. So this is a very small topic, but still there have been questions from the part. There have been questions. So the topic just talks about two kind of organism. So the first one is about turtles. Okay. So unlike human beings, there are creatures who are in the lower group of animals in who the sex of the offspring is not determined by chromosome. In human beings, it's a chromosome who determines male or female, right? But in some organisms like the turtles, and then we'll see one more example, there are snails. So in snails, turtles, like, and there are many more examples, but the ones explained in our NCRT are these two. And these are a special case. It's not chromosomal sex determination. This is environmental sex determination. So let's see what that is. It's a very simple thing. In turtles, sex of most turtle is determined by environment after fertilization. So the male and female breeds, they make the female lays egg. Okay, so the female turtle lays egg on the sand. That's how it is, right? On the seashore. So after the laying of egg, what basically happens here is the mother nature take over the conditions. So whenever the sand is almost 30 to 35 degrees Celsius, the egg has a tendency to turn out into female turtle babies. Okay, whenever the sand is having a temperature of 30 to 35 degrees Celsius, that would become, those eggs laid by the mother turtle has a tendency to become female turtle babies. Why? If the temperature of the sand is almost 25 to 30 degrees, which is lesser, right? So if the sand is more cold, that turns out to be male turtle babies. If the sand is more hot, it would turn out to be female babies. That's it. So it's purely controlled by the environment, by the temperature, by the external factor, nothing to do with chromosome. This is a special case in biology, which is where the offspring sex is not controlled by the chromosomes. And the last thing is snail. So snails are also these low creatures, low animals, right? And they have a tendency to change their sex. Here, Mother Nature is not taking part. Here, they have a tendency to change their sex even after being born as a male. So snails have a tendency, if they are in a community of more females, they have a tendency to become female or male. They can change their sex. So during the lifespan, snails, these small creatures, they can change their sex during the lifespan. Even after being assigned of one sex, they can change that sex before dying. So that's another fascinating factor of biology. Okay. So indicating, these things indicate that sex is not genetically determined in the turtle or in the snails. It can be influenced by environmental stress and factors. That's it. Okay. So this was a question which was based on that small topic we just learned. Give an example where sex determination is regulated by environment. A simple question. Or maybe they can especially ask about the turtle or the snail. Just talk about their sex determination. So if such a question is asked, you can explain either the turtle's case or the snail's case. Anyways, don't talk about humans here. Because in humans, sex determination is done by purely chromosome. That's it. So these are the topics which we had to discuss. And yes, by the sex determination, which is done by environmental factors, the topic is covered up. The chapter heredity is covered up. Let me say happily goodbye. So this is it. The chapter 8 heredity. We have covered up all the important topics and all the PYQs. I would say why I'm using the term all is because most of the PYQs were filtered and taken in this classes. So whatever topics the PYQs have come in, I have actually taken it all and presented it to you in these 3 sessions of classes. Make sure you are covering up all the 3 parts of the classes. So heredity chapter is divided into 3 parts of 1 hour each. So I hope you all enjoyed the session. I hope you all had a good journey along with us, GoScore, with the live YouTube classes. I'll be coming over with more chapters. We'll be sharing more informations in the upcoming sessions. So thank you so much for joining and showing your love throughout the entire session. So yes, this is me, Alka Rajimon. I'm signing off from GoScore Learning. Good night. Bye-bye.